So I'm going to speak about my project on biochar. Thanks for Elta introducing a bit about this. So this is a project between myself, Manoj Menon, from Sheffield, and Sylvia from York, and Masood from Manchester. And we have been working with STFC, ISIS, and IMAT beam time scientist, as well as from I-12 beamline scientist. So we were fortunate to have an industry partner, Carbon Gold, which is the leading manufacturer and supplier of biochar in the UK. So a bit of background about greenhouse gas emissions from soils. Thanks for Marcelo for introducing about carbon. So we have, these are three main gases coming out of soils, important ones. So their global warming potentials are quite different. For instance, nitrous oxide is 310 times more powerful in causing greenhouse gas effect. Globally, it is estimated that soils contribute 35% of CO2 and N2O about 53%, and up to 47% of methane is coming from soils. So this is where the biochar is coming. Biochar is known to mitigate a lot of these gases, at least a lot of literature on this. Okay. And so it's actually, what is biochar? It's actually a product of biopyrolysis. You can use any biomass. Both plant and animal waste can be used to produce biochar. Only condition is it is produced without oxygen, that is called pyrolysis. It's at very high temperature, that is around 400 or 500 degrees Celsius we use it to produce. And it's been recognized as a net emission technology by IPCC last year. So there's a scope for expanding that research. And advantage of biochar, once you turn biomass into biochar, it has a long residence time. It's almost pure carbon skeleton with a high porosity. So it can be a home for microbes for several decades or even centuries. So let me explain the difference between normal carbon cycle and biochar cycle. So normal carbon cycles, plants sequester carbon, and that is turned into biomass. And once it decomposes, it's actually going to be going back to atmosphere as CO2. But when you turn this into biomass into biochar, or it can also produce energy as well through pyrolysis, then it has a long residence time and plus additional energy can be produced. So there are lots of literature on effect of biochar on soils and how it is helping on greenhouse gas emissions. So in our project, we try to capture both direct and indirect effects. And indirect effects is actually looking through the health of soil, how it is improving the soil health. And if you increase the plant growth, obviously that's also additional carbon sequestration. And it has also been shown to influence nitrous oxide and methane and carbon dioxide emissions. So I'm going to show some output from the project. So the question we wanted to have is actually in this project we wanted to address is, does it work in the UK soils? The reasons are, we have a pretty good carbon stock, mostly as peat, but we have 10 billion tons of carbon in the UK, and which is 13% of EU carbon stock. And as I mentioned, biochar is very lightweight, high porous material with incredibly high water holding capacity. And we know our weather is not so great, so we have very high soil moisture content, and if you add more biochar, what would happen? The textbook knowledge is if you increase soil moisture to a certain limit, then it can produce more powerful greenhouse gas emissions. So this is what we wanted to achieve from this project through looking at all these gases. So some of the results I'm going to show you. So we managed to establish a network, and we carried out some preliminary imaging at ISIS and DLS. Diamond light source, and it was not enough, so we applied for beamline in October, and for 6 days, which is in that proposal we are going to do additional experiments with imaging. So we finished a very good experiment in York, and we looked at all the greenhouse gas emissions from soils, and it's still continuing. We have a MSc project and one BSc project in Sheffield. So let me show you some movies. So this is a softwood biochar supplied by the company, which is made from birch. And you can see the porosity. This is scanned at 1.8 micron resolution at I12 beamline. It's very complex pore structure you can see in there. And there are pores between particles as well as within the biochar, individual biochar particles. So I think this is the first time we ever, if anybody used X-ray imaging to produce pore structure, we can actually quantify the pore structure within these structures. Normally, we have seen images from using electron microscope, but we haven't seen tomography images like this. So you can see the differences. You can see in hardwood biochar, the porosity is not so great as the other one. Just visually comparing, we will be able to produce quantified data afterwards, but it's actually less porous. And the company itself told us that this is softwood biochar is their best product. And so they don't use so much hardwood in their products. So you can see that the pore structure is not so complex as softwood biochar. Okay, so this is just a 2D slice of the same images. Actually, you can see better here. You can see the complex pore network in softwood biochar compared to hardwood. And that is promising, so we will be continuing experiments with the softwood biochar. So coming back to the experiments we did. So this is, we did experiments with rice as well as grass. This grass is actually used for football stadiums, so the company was interested in understanding the effect of grass growth. So we decided to use grass for these experiments. So from the images, you can see that there's an increased growth in these two, which is amended with the biochar. We have biomass data, we haven't processed it yet, but you can see an increase in plant height. But if you look at the below ground situation, you can see the opposite. For instance, this is roots taken from the soils, and we scan them. So you can see without biochar, it produces awful lot of fine roots compared to with hardwood or softwood biochar. This is perfectly possible because plants don't have to produce a lot of roots if there is plenty of resources available or water available. So in fact, roots tend to forage quite a bit if there is not enough resources around. So I think the hypothesis here is actually biochar might be giving enough moisture, so the roots, they don't produce a lot of roots. So coming back to the CO2 capture. So here you can see that in presence of plants, in both biochars is actually sequester more carbon. This is mainly through the increased plant biomass we have seen. And without biochar, of course, it's not sequestering carbon. But the opposite can be seen when there is no plants. So if you add biochar straight in the soil, it can slightly increase the emission of CO2. But this flux is very small compared to that one. So it's only 100, 150 or so. This is one order of magnitude higher. So there is a benefit of having plants in place with biochar. So obviously we have a lot of data to process. We haven't looked at the other gases yet. And also this data is only for 2 days. We have continuously monitored throughout the grass growth. So there is a lot of data to process. And we have a lot of soil analysis pending. And yeah, hopefully we get a paper. Thank you very much.